Good afternoon, everyone, and thank you for coming to this session. And I think it's a really great position for me between Dr. Li and Dr. Song, as my work highly related to both of their work. And today I want to talk about a very specific TMS treatment and what's the potential. What's the possible mechanism behind? Because we use TMS for many years, and it has been expanded from depression to PTSD to obsessive compulsive disorders, but the exact mechanism behind is still a question. So before I start, I have nothing to decline. So today I want to cover these 4 areas. First is the glutamate system and the depression, and second is what's the effects of this new treatment on the biological system that we are using to test the hypothesis, and then we will move on to the possible mechanism after, and by the end we'll come back to the clinical part and to see what can we use, how can we use this different approach even beyond depression. So we all quite familiar with serotonin hypothesis and norepinephrine hypothesis for depression, but there's definitely beyond that there's something new that's happening, and I think that later on Dr. Song may bring more details about this. It's about the glutamate system. So we can see from this side, and depression is such a complicated case that it's involved from the stress, from the environmental impact, to the genetic components, the vulnerabilities, and even to the medical conditions that when we treat the patient, we all know they're all very high risk factors, and it's influenced by hormones, and down to the very important part is inflammation. So that's put depression in the center to connect from the environment to the biological system. So what things actually has put them together? It's still a really big question here. So, but as Dr. Li said, now the new theory suggests that depression is a disconnectivity disorder, which means that the brain functions between different areas, they're not working properly. As we all know, that our prefrontal cortex is the center for executive function. It helps us to make sound judgment, to regulate our emotions, and to have a proper social interaction. When it's become less active and less accurate, and our lower function center, which is like limbic system, hippocampus, amygdala, which some people call dinosaur's brain, start to take in control. That makes us a very big bias towards the signals that we're receiving from the environment, and then make very emotional decisions. That's definitely will influence our function. So how do we treat it? I think the new theory, as Dr. Li mentioned, is to rebalance that, to reconnect the different areas to make the function properly balanced. Our CBT is one of the perfect example. CBT helps us to pay attention to the details, to the objective and balanced view. Then we can use our cognitive function to regulate our emotions. So how does that link to the NMDA? We all know that from our medical school biology that the NMDA is an excitatory neurotransmitters. It's one of the most important information delivery system in the brain. It helps us to learn. It helps us to make really important decisions, and it also helps us to handle the very complicated cognitive function. So from this review, we can see that how does that affect and become part of the depression system. One is when the animal got stress, it's causing the cortex or the cortical steroid system, and then to start to impact on the neuroplasticity, and leading to a very leading to a decreased neuroplasticity and also decreased connectivity. It's not only affect the neurons, but also affect the glial cells, have a decreased numbers of glial cells, and decreased connectivities as white matter plays a role. And also, it's not only the one way to explain that the excitatory neurotransmitters play the role, because if we have too much excitatory neurotransmitters, it becomes toxic. We need the inhibitory one to balance that. But the stress actually leading to more excitatory, and surprise, the inhibitory is the GABA. So the imbalance of these two leading to a this is a serious thing, leading to a synaptic disconnectivity. So from this one, we can see in the patient with depression, the glutamate is too much, and the GABA is too little, which is really a simple and overly simplified case. So if we want to treat that, we may want to balance them back. So now we talk about RTMS. We all know that TMS was originally developed in 1911s. And I don't know who want to put the head into this one for these days, but they're really brave. I gave them the credit. So, and later on, there's tons of new equipment has been developed, and all of them are highly focused on the accuracy and the intensity in terms of the treatment. But what I want to talk about today is a new equipment which totally different from the previous one, because all the previous one need to actually activate the neurons to create an active potential, which means that the neuron need to be excited to pass along the information. But this new equipment, which have a we call the subthreshold stimulation. That means with this treatment, and neurons won't be excited. But how does that deliver the function is nobody knows. So what's the benefit for this is 2 days ago I gave a presentation about the clinical evidence to show how fast this treatment can improve the treatment-resistant depression and also bipolar depression. It's like 3 or 4 treatment can significantly improve that. And we can see it's very portable and very comfortable way that patient can sleep on, and it's really affordable, and it's very rapid improvement. So this may change our treatment options and also the social system to help us to reduce the cost. That's why I really interested for the studies. So just like RTMS, we do not know what exactly happening in the brain. So we take a liberty to try to explain it and to fit into the current theory. So the first one that we did was we used a stroke model. We borrowed a stroke model because we all know that when the animal or humans with a stroke, there a huge release of excitatory glutamate and to make we call it excitotoxicity. So we generate the model by block the vessels and to see how does the treatment as a prevention and ongoing treatment after 3 days, see what's happening in the brain. So it's very interesting. This is the electrophysiology studies that we did. What we did is we just put a probe into the hippocampus, which can help to record any actions that are related to the excitatory neuron activation. So we can see the red one is what the animal is supposed to react. After we gave a chemical to induce this action potential, it can quickly go back to the level. So in this case, it does not have a prolonged activation. That's prevent us from excitotoxicity. But when the animal have a stroke, and we can see that in the stroke side, and the activation actually continues and lingering there. So that's one common agreed mechanism in terms of the cell death after one of the type of cell death after the stroke. So when we give the treatment, it's very interesting. We can see the treatment itself actually can help to modify and to normalize the curve. You can see here, it's a bit small, so you can see the green one, and it actually have a very big difference between there. So that means the excitotoxicity can be blocked by the treatment. And also, it also reflects on the cells, and we can see after the treatment, the neurons in the hippocampus area actually being protected from the excitotoxicity-induced death. So, and it's very interesting that another group independently did the same work, but on an animal model of Alzheimer's, and they have the similar results like us. So that means this data was repeated independently by the other people. So this is one area we explored. And the second one that I want to go back to Dr. Lee's work is about oligodendrocyte dysfunction. We know that it's also being well mentioned, and the model has been well mentioned. So what we did, we used the same model, and then we use the treatment and then to see how does that affect the oligodendrocyte. And surprisingly, that it helped to prevent the oligodendrocyte death from the chemical-induced toxicity. And the toxicity is also highly associated with the glutamate activation. So, but we don't know if this is a secondary outcome because we protect the excitotoxicity, we prevent the excitotoxicity, so the oligodendrocytes get survived, or it's have an act directly on oligodendrocytes. So we took advantage of a cell culture system, which this CG4 cells is an oligodendrocyte type cell. It cannot survive by itself. It has to grow in the medium that's created or secreted by the neurons. That means they have a crosstalk. Neurons support oligodendrocyte. So what we do is we treat separately the oligodendrocyte, the CG4 cells, and the support neuron cells. And then we can see what exactly happening. And our data actually, this one, I didn't update the new data. Our new data showed, actually, it's very interesting to see, there's a synergetic effect. That means if we treat both type of cells, it gives a better effect. That means the beneficial effects on oligodendrocytes may not necessarily solo independent events. It's also supported by the oligodendrocyte, by the neurons function. So we all know that oligodendrocyte is not just a passive cell that's to support neuron or to protect neuron. It's linked, have a very complicated interactions with neurons. Neurons that's wrapped by the oligodendrocyte actually give the information to tell the oligodendrocyte where I need you, how thick the myelins I need you. So in that case, they have a synchronization. So in this case, we can see when we have activity-dependent myelation, that means neurons will tell what to do to the oligodendrocyte, what to do, and glutamate plays a very important role in the directions. So that change may also involved with the glutamate system. The third one is inflammation. We know that no matter what kind of brain, currently all the brain neurodegenerative disorders and depression and schizophrenia, they all have inflammation involved. Microglia is one of the most important cells in the brain for in responding to the inflammation. And Dr. Lee showed this data that after treatment actually it's decreased the oligodendrocyte or the microglia activity. That means it can decrease the inflammation. This is also highly associated with glutamate. I'm going to skip that data. And recently I did a very different research and using the same treatment. So what we did is we generated a traumatic brain injury by drop a weight on the rat's brain. And then after that, we can see the behavioral abnormalities like weakness and cognitive impairment. And one of the very interesting part is we found it actually can disrupt the circadian rhythm. We all know circadian rhythm is such an important thing for psychiatry practice. I think 80% of our patients have a problem with sleep. And the light therapy is one of the most evident in terms of treatment. So circadian rhythm disruption is super important for the mood regulation. So by that way, we want to see what does this treatment do with the circadian rhythm. And surprisingly, we found that it actually can regulate the genes that are critical to maintain the circadian rhythm. And I didn't have the data here, but actually all those genes are regulated by glutamate system. So in summary, even that we have very individual evidence to showing that oligodendrocyte protections, microglia dysregulation, de-inhibition, and also the circadian rhythm regulation. But if we can trace back, we find that almost everything go back to the glutamate. So the theory we were thinking is the oligodendrocyte, the glutamate system is the key, probably shares is a shared mechanism. So why this treatment can change this? And one of the theory is when I applied this treatment, I tuned the frequency to called 40 hertz, is a gamma oscillation. So we all know that gamma oscillation is super important for higher cognitive functions. It's generated by the balance between excitatory neurons and the inhibitor neurons. So you probably heard about one called the PV neuron. PV neuron is the most abundant inhibitor neuron. It's also happened to be the most abundant myelinated neuron. So in schizophrenia, in Alzheimer's, and in bipolar and stroke, this inhibitory neuron function all got decreased. So, and interesting that there's two papers published by MIT for the last 2 years. What they do is it's quite genius one, that once they flash the light at 40 hertz. So then they find that it can decrease the inflammation and also the plaques that we supposedly contributing to the Alzheimer's. And later on, the same group used sound at 40 hertz. They found it's even have a better effects on it. But that's why I tuned this treatment at 40 hertz. So what's the benefit here is we do not need to put patient into the unpleasant situations. And also the penetration are much more global. So that's why I hope this will be a better treatment and based on that mechanism. So finally is I mentioned about bring them beyond depression is we can see that multiple sclerosis, TBI, stroke, all those neurological disorders share the same common pathways, neuroinflammations, activation of microglia, and metabolic disturbance, oxidative stress and excitotoxicity, oligodendrocyte damage. So all that, and also all the neurological disease also share the symptoms like cognitive impairment, mood disturbance, sleep problem. So that I bring them to see this treatment may not only for depression, but all probably will target on more broad areas. So this is my team, and with their great help, and also with the collaborations with other great scientists, that made this work happen. Thank you very much.